Hello, everyone. I hope you are doing amazing. Today, we will be looking at, this is for my biology class, and so we're going to be looking at macromolecules and enzymes. So, macromolecules, well, what does the prefix macro mean? Macro means larger as opposed to micro. And there is this kind of what's called a biological hierarchy. First, you start with the atom, then you increase to the molecules, and those molecules make up macromolecules. And what comes after macromolecules is the cell. And I think there's a pretty good chance we'll talk about that later on, but today we'll be focusing on macromolecules. And with all of these things biologically, they're all made of a element that is present in every living thing, that is carbon. But macromolecules are always made of three elements: carbon, hydrogen, and oxygen. And as we go on, they will have different elements. And there are four types of macromolecules: one, carbohydrates; two, lipids; three, proteins; and four, nucleic acids. Now, before we continue, there's something very important to understand that is monomers and polymers. Monomers make up polymers. Now, what exactly are monomers? They are very small molecules that make up bigger molecules, and those bigger molecules come together to make the macromolecule. But this macromolecule can is a kind of general term of what a polymer is. See, there are four different types of macromolecules, but monomers make up polymers, and those polymers are within either a carbohydrate, a lipid, a protein, or a nucleic acid. And so, just for example, nucleic acids have two different polymers. Now, usually they have all kinds of different polymers, several dozen, but nucleic acids have only two, and that is DNA and RNA. So DNA and RNA is made up of nucleotides, which is the monomer of a nucleic acid. DNA is a nucleic acid, RNA is a nucleic acid, but they are types of that general term, which is a type of macromolecule, which is nucleic acids. Okay, so we'll go into carbohydrates. Well, what exactly do they do? Well, first off, we know that they're made of carbon, hydrogen, and oxygen. It's in a 1-to-1 ratio, not like 1-to-1, like one number two number one ratio, as all macromolecules are, with the exception of lipids. But they are made of just carbon, hydrogen, and oxygen. Their primary function is to provide quick energy. And you can see how that would be unhealthy. There's this very unhealthy connotation behind carbohydrates, and that's because they provide quick energy, and too much quick energy is unhealthy. And of course, this is where you get your sugars from. Every kind of sugar and chemical form, glucose, fructose, starch, they all are carbohydrates. Pretty much anything that you hear that ends with "ose" is a carbohydrate. Now, there are two kinds of polymers that are carbohydrates that do not end in "ose," and that is starch and glycogen. What is the monomer of a carbohydrate? It is called a monosaccharide. A monosaccharide. And there can be, and this applies to all of the other macromolecules, but it's specifically important to carbohydrates, and that is a disaccharide. And by just knowing the prefix di, that is just monosaccharides bonded together. An example of this is sucrose, which is glucose and fructose bonded together. And sucrose is still a polymer. It is, even though it's two polymers combined to make a chemical, it is a polymer that is a carbohydrate. And of course, whenever you get enough of these monomers, they make polymers, right? Monomers added together make up polymers. So there's another term. You have monosaccharides, which are the just the monomers of of the polymer that is a carbohydrate. And if you have two monosaccharides together, it makes a disaccharide. And of course, if you get enough of these monomers, which are monosaccharides, they make the polymer, and this is called a polysaccharide. And of course, again, we won't go into the exact terms and the different monomers, and we will go into the monomers, but we won't go into how they add up and the building blocks specifically of each kind of macromolecule. But it's important to know about carbohydrates. Although carbohydrates do primarily provide quick energy, they also make up cellulose, which makes it's used in the formation of cell walls. And again, macromolecules, what comes after a macromolecule, that is the cell. And cellulose helps to make the walls of those cells. Onto lipids. What are lipids important for? Well, first, what are they made of? They're made of the same thing, but with a different ratio. They're made of carbon, hydrogen, and oxygen, except there is a exceptionally low amount or number of oxygen, and this is the only exception to the 1-to-1 ratio. But what is their purpose? Their lipids are important for energy source, protection, and insulation. Not an energy source, energy storage. So, we think of people who might be overweight, they will have a lot more fat. And that fat, it's a kind of storage of energy. After eating so many lipids, that fat can be combined into energy. So that's why you lose a lot of fat if you don't eat for a while because your body needs energy, right? So it breaks down the lipids, those macromolecules, and it uses it for energy. It's a very productive and efficient way to store energy. They're also used for protection. So, uh, one of the traits of living organisms is organization. And so, lipids are organized to be used as a kind of protection. And maybe not so much with humans, but it's used for organ protection. Maybe the way we think about lipids or fat, we think, oh, like protection on the outside, but mainly in humans at the very least, lipids are used to protect organs and insulation. And again, maybe not so much for humans, but we take in like walruses, or I think walruses, maybe seals. They have blubber, and that blubber is made of lipids. And this helps them be very insulated. Or whales. Whales have blubber, and they're in very cold waters. And how do they not get super cold and mess up? It's not all this fancy stuff. It's lipids. Lipids insulate them and keep them from being cold. So what makes up a lipid? What are the monomers of a lipid? And usually each macromolecule has only one monomer, but lipids have two monomers. Those are fatty acids and glycerol. So fatty acids and glycerol, they are monomers which make up the polymer, which is lipids. And lipids have a pretty cool characteristic. They are insoluble in water because they are nonpolar. So this is pretty important for leaves if they don't need water. They have a certain kind of lipid which puts like rejects water or repels water away from their self. So in the same way that there's monosaccharides, disaccharides, and polysaccharides, there is saturated, monosaturated, mono unsaturated and poly unsaturated. And so there's double bonds, right? It's kind of hard to explain. I might put a picture up, but all you really need to know is that if fatty acids are not saturated, they don't have any double bonds. If they're mono unsaturated, they have one double bond in a singular fatty acid molecule, right? And if they're unsaturated, they have multiple double bonds. Moving on to proteins. They're important. They are probably the most important macromolecule. They each have very significant roles. There's only four types, right? And they're all for organisms. But proteins are very important just for construction and building. They build cellular structures. They build microtubules and enzymes. And enzymes are very important. We'll talk about that later. They build organism structures, your nail. It's a collagen, and it sends chemical signals and messages. So every single chemical reaction in every organism is sourced, it is energized by proteins. Really enzymes, but proteins make enzymes. So instead of just being composed of carbon, hydrogen, and oxygen, it's composed of carbon, hydrogen, oxygen, and nitrogen. What's the monomer of a protein? Amino acids. There are 20 types of amino acids, and they make up thousands of different types of proteins. So think about all the combinations you can have with just 20 different individual things. They make up one protein, or they make up thousands of different types of proteins. So each monomer is connected by something, but it's important to know what connects amino acids. And what connects amino acids, which are the monomers of the polymer, which is protein, the thing that connects the monomer, which is amino acids, are peptide bonds. And these amino acids can be placed in a different order or different sequence or in different numbers, and they can make these different types of proteins. That's how they're made. As a result of this, we have certain monomers that make up the polymer, and then we can build up and see which building blocks are called which. And so, same way you have monosaccharide, then disaccharide, and then polysaccharide, you have just polypeptide. That's in the same way you would say polysaccharide, there's polypeptide for protein. So just to show you, like you've probably heard of these are examples of proteins. They're very important. Hemoglobin, in the blood, antibody, enzyme, and polymerase. I don't know what that is, to be honest. The fourth kind of macromolecule is called a nucleic acid. Nucleic acids are made up of five different kinds of elements, as opposed to carbohydrates, lipids, and proteins. So they were made of CHO, carbon, hydrogen, oxygen, and then we got to proteins, which make up carbon, hydrogen, oxygen, and nitrogen. And nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So they make up here's an easy way to remember it. There is four different types. If you go to carbohydrates, it's just CHO, carbon, hydrogen, oxygen. You go to lipids, same thing, CHO. Protein, that's nitrogen instead of those three. And so you make CHON. And then you get to nucleic acids and you make up CHOMP. Not CHOMP, but CHOMP. So what's the point of a nucleic acid? A nucleic acid stores and transmits genetic information. And as we discussed earlier, that's why the polymers of nucleic acids are DNA and RNA. They carry that genetic information. So what's the monomer of a nucleic acid? It's called a nucleotide. Nucleotides are made up of three things: a nitrogenous base, simple sugar, and a phosphate group. And of course, the polymer is DNA. So we'll kind of touch what DNA is. What is DNA? It is a master copy of an organism's information code. It carries the code. It's a copy of you. It is the thing that physically makes you separate from every other human and every other animal. So it contains the instructions used to form an organism's enzymes and structural proteins, which is what makes you up, right? So the proteins are literally like you're the building block of everything that you are. And of course, every macromolecule is just as important. Like if you didn't have carbohydrates, you die. You didn't have lipids, you die. You didn't have nucleic acids, you die. Um, but proteins make up primarily of who you are, and nucleic acids, they remember what you need, right? So think of people with lactose intolerance. They lack a enzyme, which is a kind of protein, which is called lactase. And lactase breaks down lactose. So if you can't break down lactose, you have all those issues, right? But because they lack lactase, which is a kind of enzyme, they lack it, and they will always lack it. They might change for the mutations and stuff, but they will always lack it because they lack lactase, which is part of their genetic code, right? In that case, what is RNA? RNA is a copy of DNA. It's funny because DNA is a copy of genetic code. So RNA is essentially a copy of a copy of your code. So why don't you make a copy of a copy if you could have DNA nonstop? Well, it helps to send signals way faster, and it is there when the proteins are being built. So it's like the DNA is like the supervisor, and then the RNA is like the boss. And so the people aren't always going to go like the construction workers aren't going to go to the supervisor nonstop. They go to the boss and they say, "What do we do?" And the boss tells them what to do. Well, how does the boss know what to do? From DNA, if that makes sense. So moving on to enzymes. Enzymes are, of course, a special kind of protein. It's called a specialized protein. It is a biological catalyst. What does that mean, catalyst? It basically means that it breaks down other macromolecules or just other molecules much quicker. So an enzyme, it's kind of complicated. I put a diagram up, but enzymes have, they're kind of like a Pac-Man. Think about like Pac-Man, right? And they kind of have the mouth. What in the mouth would be called a active site, okay? And it's just called an enzyme. So the Pac-Man part is called an enzyme, and the mouth is called an active site, and something needs to go into the mouth, and that's called a substrate. So basically, whenever the substrate is in the enzyme, it can do that catalyzation process. But if it does not, it can't. It's kind of useless, but it's not inactive. It's actively searching for, and it happens very rapidly, of course, it's actively looking for substrates. So to go more in depth of what catalyzation really is, whenever macromolecules are being broken down, there's something called activation energy. There's a certain threshold for energy that is required. What enzymes really do is they decrease the energy that is needed for the activation of the breakdown of that macromolecule. And there are many different types of enzymes. So basically, what happens is amino acids make up proteins, but sometimes amino acids make up enzymes, and enzymes are still proteins. But you gotta remember, the enzymes are still made up of a certain combination or sequence of amino acids. And each of these enzymes have a specific function. So, for example, like I said earlier, lactase is an enzyme that breaks down lactose, and lactose is a carbohydrate. Without lactase, you can't break down lactose, and boom, you get a lactose intolerance. So if you wanted to be a nerd like Jimmy Neutron, you could be like, you're not lactose intolerant, you're lactase deficient. So then there's two things about this diagram, like the Pac-Man diagram. It's not actually what it's called, but they're reusable. So whenever the substrate enters the active site, they can use the substrate in a way, and it'll make a certain result that breaks down whatever macro molecule or whatever thing needs to be broken down. And right after that, they can go and find another substrate that they need. And it's a specific kind of substrate. The enzyme is built to have a specific kind of substrate within it. If it has any other kind of substrate, it won't work. Literally won't be able to go in. Kind of like a key and a lock. A lock needs a specific, like a very specific thing. It has to be that thing. It's not different types of keys. One specific kind of key. You can duplicate it. You can have all kinds of different kinds of that key, but it needs to be that way. And it's the same way with enzymes. Enzymes need a specific substrate to have the catalyzation process. And so enzymes are really affected by two things. So there's a sort of amount of activation energy, or their energy. It's not activation energy, but their energy, their ability to break down other macromolecules. And so it's affected by two things. One, temperature. Two, acidity. That's how you pronounce it. Enzymes have a optimal range where they are the most active. Below that range, it becomes inactive. It just won't do anything. Above that range, it will denature. So a enzyme can go below its optimal range. It won't be able to do anything, but it can go back up to the temperature and it will be active again. Whereas if the temperature were to go above the optimal range, then it would no longer be effective. It would begin to unravel or denature. Now, this isn't actually what happens, but think of it as if the, the lock and the key thing, that the lock, the enzyme, is melting, that it is starting to deform. And after that, you can't put the key in, right? It's completely messed up. The shape is messed up. You can't do that. So whenever this happens, it's called denaturing. It's it's unraveling. The real term for it is denaturing, but it's unraveling. That's another way to put it. And it loses its function. The enzyme shape changes, and the active site, remember the kind of mouth of the Pac-Man or the lock, will not be the correct shape for the substrate that is needed for the enzyme. It's not like it can change shape and other substrates can go in it. That can't happen. The second thing that affects enzymes is the acidity. If acids are, well, acidity, but if it is less than 7, it's called an acid. If it's greater than 7, it's called a base. If it's neutral, it's just neutral. Sorry, if it's 7, then it's neutral. So here's how it works. There's something called enzyme concentration. A higher concentration of enzyme will result in a faster reaction rate if enough substrate is available. So substrate is kind of like the fuel that it gets its concentration from. So then there's substrate concentrations, basically how much can be taken from the substrate. As substrate concentration increases, the reaction rate increases until the active sites are saturated, at which point the rate reaches its maximum. So here's a little summary of macromolecules. Kind of carbohydrates are organic molecules or macromolecules, and their building block is a monosaccharide. What is their function? They provide quick energy. They also make up cellulose and everything that is responsible for making cell walls. What makes up a carbohydrate is carbon, hydrogen, and oxygen. What are some examples of carbohydrates? Glucose, fructose, starch, glycogen. Lipids, what is their building block? Lipids are made up of fatty acids and glycogen. What is the purposes of lipids? Lipids provide protection for organs or maybe on the outside. They store energy in the long term, not like carbohydrates. They have very short-term energy. Lipids store energy for a very long time, and they can be broken down back into energy. And they have a third function, which is they provide insulation. Like I talked about earlier, whales have blubber, and blubber is made of lipids, and that lets them stay warm. What are the elements of lipids? Same thing as carbohydrates, carbon, hydrogen, oxygen, but a very low amount of oxygen. What are some examples of lipids? Fatty acids, example number two, and example number three. Proteins. What are the building blocks of proteins? Amino acids. What is the function of proteins? Pretty much every single building thing of your body. But the three main ones are they send chemical reactions, they make up what is it, organism structures, they make up hair, nails, all that good stuff. And they make enzymes, which are like super duper important. Nucleic acids. Oh wait, no. What are the elements of protein? Carbon, hydrogen, oxygen, and nitrogen. For some examples of proteins, enzymes, hemoglobin, antibody, you name it. Nucleic acids. What are the building blocks of nucleic acids? They're called nucleotides. What are the functions of nucleic acids? They store information. So in the same way that like lipids store energy, nucleic acids store information. And they have a kind of master's code of the genetic information that we have. What makes up nucleic acids other than building blocks, like what are the elements? Carbon, hydrogen, oxygen, nitrogen, and phosphorus. What are some examples of nucleic acids? There are only two, DNA and RNA. That is the building blocks, functions, elements, and some examples of every type of macromolecule. And to give a brief summary of enzymes, enzymes are a type of protein, and they help a lot with catalyzation of different chemical processes, and they're pretty important. And that's about it. I love you all. Hope it was a good blessing, and God bless you all. Have a good one. Goodbye.